be quite important. I've been looking at mammalian lung development, which is different from avian lung development. The mammalian lung, of which this is a plastic cast of an adult airway, and it's omitting the blood vessels, but the mature lung is beautifully optimized for efficient use of materials, efficient transport. It's not actually as efficient as the avian lung, but within the constraints of having a dichotomously branched system, it's terrific. And your typical math reaction is, it's a fractal, I know how to model fractals, but it's really not quite as simple as that by any stretch of the imagination or by any stretch of the tissue. In particular, it's developing not just with the airway, of which this is the lumen, a cast of the lumen, but with all kinds of tissues around it, including blood vessels. The embryonic period, when it's branching, this is a mouse lung, which grows pretty well in a dish. In the embryonic period, in the early stages, it pushes out from the tube, which becomes your gut, and branches, and that branch branches, and so on up to, in the human, 23 generations. And it's all, interestingly, it's for the most part happening on a consistent length scale. You notice the tubules are not getting to that nice cascade of length scales that you have in the mature lung. That's going to happen later. The branching process is all at roughly the same length scale, and the tubules remain for a long time at roughly the same length scale. So there's something else going on here. You've got multiple tissue types. So this here is the lumen, and you've got a lumen from the get-go, as far as I can tell. I think it's hollow the entire time. It doesn't form a lumen. And you get an extension of a bud of epithelium into a mesenchymal tissue that's not terribly well differentiated at this point. There is a mesothelial surface on the outside. In the older tubules, there's some smooth muscle, which is going to come in pretty soon in my talk. This is going to be the focus of the talk. It's something that people who don't think about lungs don't really think about terribly much is, remember, this is inside its mother. It's filled, and this lung is filled with liquid. It's not filled with gas. So it's a mechanically very different environment. You don't think about it, but then in retrospect, of course, it is filled with liquid, not with gas. So one of the main questions, there's a lot of questions here. One, how does the branching occur? Two, how does mechanics come in here? Three, how do these ducts go from all pretty much the same diameter to what we saw on the previous slide, where the earlier generations are much bigger and optimized for efficient use?
What's the temperature?
This?
Yeah.
2 days, I'm guessing. It's about 2 days in the mouse. We know that there's a lot of mechanical input into this system. And it would be nice to understand what's going on. It's like most developing tissues, there's a lot going on mechanically. And in the normal environment, for example, it hasn't been widely studied. In fact, the only news I know of it, of these tonic contractions, are unpublished data from a guy at Princeton. A tonic contraction meaning the whole lung is contracting and holding and then releasing. There are fetal breathing movements later on, just as once you've got muscles, you start kicking. Once you have a diaphragm, you start breathing, even though there's no air. It's practice. And we know that if that's disrupted, that the lung doesn't develop very well. Similarly, as soon as you've got smooth muscles, I think in practically any tissue, as soon as a smooth muscle develops, you've got some kind of contraction. And in this case, there are peristaltic waves in the airway. And it is known that if you disrupt the peristalsis, it interferes with lung development, just as if you interfere with the breathing movements, it interferes with development. Bad things can happen. So a not terribly uncommon birth defect is if there's a hernia in the diaphragm, it can lead to the internal organs not giving the lung enough room to grow, and that interferes with development. That seems to be too hard a system for me to even look at. And the fetal breathing movements are not during the period when the branching is occurring, so that's of less interest to me right now. I'm mostly interested in what's going on during the period of branching, and the airway peristalsis certainly starts when the branching is occurring, and it keeps going. Experimentally, people have looked at occluding the lung, just blocking up the trachea, or when you're growing it in a dish, make sure that the end is closed, either by cauterization or some kind of plug. There have also been experiments in late fetal sheep where they will surgically implant a catheter into the lung to drain the fluid and see what happens. So we do know something about many of these mechanical inputs, but we don't really know what, we don't really know why the lung growth would be enhanced or inhibited by these kind of gross treatments. And I'd like to kind of get a sense of when you're doing something, such a large mechanical perturbation as blocking the trachea, for example, or blocking peristalsis, or even allowing peristalsis to occur, what is that doing mechanically to the tissues, and how could that be enhancing the development of the lung? And in this talk, I'm going to focus on just the two aspects of airway peristalsis, which you saw in the movie, and experimental occlusion, because we know quite a bit more about that system. And I don't want to look at that whole big moving lung. I want to simplify as much as possible so that we can learn as much as possible from the simple models before diving into everything we think we know. And so just to simplify the histology, I'm just going to consider a simple epithelium, smooth muscle, mesenchyme, and of uniform properties, a uniform lumen, and the surfaces that connect them. So the pleural surface, there's stuff going on at the mesothelium, but it's just going to be simplifying the histology. So in the case of airway peristalsis, we need all of those regions. What does it look like? So this is early in development when the smooth muscle has formed. The peristalsis is not quite coordinated. I don't have a video of peristalsis later on when it really is very robust. So this is kind of in random places, and it's just a light squeeze.
Sharon, just for curiosity, how is this done experimentally? Because it's certainly not in the mouth.
It does it by itself.
Sorry?
It does it by itself.
No. Do you explant the lung tissue from the embryo?
Oh yeah,
Okay.
yeah. And then it grows. I don't do these experiments. I'm reporting other people's experiments. But it does grow pretty well. Usually they will do some kind of moderately flattened culture for microscopy. But not everybody. There's some people using microfluidic chambers, so they'll keep a full 3D shape.
And the time scale of this peristalsis? Is this real time? Or
Is it real time? So I think this is a little bit sped up, but not much.
Thank you.
So it's in tens of seconds that a wave would propagate. And it depends on stage and so on. Yeah.
What exactly are we looking at here?
This is a mouse lung in a dish that is undergoing peristalsis. And this is kind of a trial version of peristalsis. This is not robust. I unfortunately don't have a video of a mature form of airway peristalsis. So this is kind of in random locations and random directions. Which way do you think it would go? Okay, just your biological instinct, which way do you think the peristalsis would go as a habit once it develops, once it's really synchronized with itself? This way.
Getting stuff on the wall.
It goes this way.
Yeah, of course, but I mean, if I assume that there's some central pacemaker, it must be here for symmetry.
That's an argument.
Yes.
That's an argument. Yeah, okay.
Can I ask a question? Is there smooth muscle, are there smooth muscle cells around every bit of that?
No.
And only
No.
some of them are contracting in this video?
The smooth muscle develops first. It affects as it's developed, as this thing is elongating and branching. It starts at the lowest generations, which are the most differentiated. You have more tissue types in the older tissues. And then the airway, as it's spreading and branching, the differentiation proceeds also outwards from the central location. So the smooth muscle is going out here, kind of out to here, not in the very end.
So most of the smooth muscle that is there is participating in the contractions that we see.
Yeah, depends how well developed the smooth muscle is.
I mean, in this particular case.
Yeah. I don't know. It has been pretty well studied. There is a surprising number of papers on airway peristalsis where they've quantified the waveform and so on. It's pretty nice, actually. But it is surprising. It was surprising to me to realize that, in fact, the wave goes this way. Maybe it was not a surprise to everybody, but that is amazing instinct, actually. But it's not too clear. Well, maybe it is. I don't know. We'll get to that. This is actually not my favorite part of this talk, but it is surprising. Now, the other thing, the static stresses are actually my favorite part. What does it look like when you just stop the film? It can be a huge amount of occlusion. You can, even though this is not a very well-developed waveform, the occlusion can be complete. It's just a matter of the contractile force relative to the stiffness, relative to whatever it's resisting. But a complete occlusion that moves distally is going to cause a large blowout of the tips. That's got to be that's got to have quite a mechanical input to those tips. There's a lot of pressure there, presumably. And what is that doing to the morphogenesis in these tips? We know that if you block it, you get less robust lung development. What's it doing, in fact, to the morphogenesis right next to the occlusion? It presumably would be mechanically stimulating right near the occlusion, as well as far away, not necessarily in the same way. So I thought this needs a bit of mechanical analysis. And there's just so many what-ifs here that I thought formulating hypotheses is just absolutely pointless without some kind of quantitative framework.
So
Yeah.
I didn't entirely get it. I mean, this is a frame in the peristalsis movie, right?
Yeah. Yeah.
But then I don't see why you say complete occlusion cannot, like, because I mean this is it's moving peristaltically, so the fluid is moving distally, right? So
Yeah.
then maybe I don't understand the point of what you're trying to say in this slide. What I'm
just trying to say that the tips there's so much pressure when it occludes completely that notice this one is larger than this. These green rings are the same, or scale bar.
So maybe what I don't understand is why should it be complete?
It just is.
Oh, okay.
It just is.
Oh, okay.
But my question is, what is this doing to the morphogenetic system? Because we know that if you block the peristalsis, you don't get good lung development.
Sorry?
We know that if you block peristalsis, like a nerve block or whatever,
Yeah.
you don't get good morphogenesis. And so what's going on mechanically when this thing is happening? So, there's a lot of hypotheses. You can say, okay, so probably the cells are being stretched. Are they being stretched the same in different places? Stretch has direction. Is it longitudinal? Is it circumferential? And so on. How does the stiffness of the tissue affect the stimulation and so on? So we did a lot of different angles of a mechanical analysis of the of airway peristalsis. It was mostly work by my grad student, who was a mechanical engineer named Kishore, but not Musliganti, Kishore Boka, who did most of the work. This is another view of the tissues in the embryonic lung, just to illustrate that this is the mesenchyme, and you've got these tubules, which are a simple epithelium, and you've got these nice clear lumens. But this is really too much information here. I want to just for simplicity, consider everything as a continuum, because I don't know how to work with this much information here. I want to simplify the geometry as well. That big branched structure is just too much to think about, so maybe we can just think about what's happening in one tubule with one end. Because even that is a big task, to understand peristalsis to a tubule with an end. There's decades and decades of analysis of peristalsis, some of it done by people in this room. And it's all with infinitely long tubes. I have yet to find anything where the fluid is being pushed to a closed end. So even that is worth looking at. And so let's simplify the geometry to a straight tubule. Let's simplify the histology to just a few tissue types. Let's simplify the mechanics to just elastic materials. And the lumen fluid, well, we don't know anything about the lumen fluid. What is the fluid in the lumen? Don't know. I mean, people have looked at the lumen fluid of near-term fetuses, but not the embryonic fluid. We don't know what it is. And let's consider even the way the cells are packed in epithelia really simply. And there's a lot we can say, and I want to actually rush through what we've learned, because this is not even the most interesting thing I want to talk about. So, you can keep track, you can estimate where the fluid is being pushed to by conservation of volume, and that gives you an estimate of how the diameter of the tubule is going to be changing. It gives you an estimate of how much the tissue is being stretched.
By how much is it stretched?
A lot. I mean, a lot. So if you get complete occlusion, and it goes a long way, it's going to stretch everything in front of it. It's going to push all of that volume as far as it can go until it stalls, or until you run out of smooth muscle, right? And I also want to ask, so what is that doing to individual cells? They're presumably being stretched. How much and in what direction? So just in terms of also simplifying the histology and the mechanics, let's just think of this as like plastic. That is, it can be divisible, just an elastic material where each cell is just a portion of it, and just do the geometric analysis of when you're stretching the lumen, how much are you stretching the cells? And just, this is a geometry test. This is something you can assign to a smart high school student to figure out the conservation of volume in these solids. So if you're packing wedges into cross sections of a tubule, and you completely close that tubule, then their shape changes. And how much does it change? Well, it would change a lot. If you consider the end to be a hemispherical cap and you're packing hexagons, well, it's just a geometric conservation of volume question. And if you blow out the tip by a doubling of diameter, depending on the radius of the lumen, you get huge changes in all kinds of measurements of the cells. If you're compressing the stalk to complete occlusion, you're getting huge changes in the dimensions of the cells. So there's a lot of stretches and compressions in the different directions, in the different locations, and what's going on in the stalk is quite different from what's going on in the tip. So biologically, you have to be thinking about the stimulation of the tip and the stalk separately, because one is being stretched and the other is being compressed. Okay? One thing learned. What about inside a cell? Again, a really simple analysis. Oh, this really doesn't show up terribly well. The colors. Well, so this is again a simple elastic analysis. If this is a uniform material, although I made the nuclei, we made the nuclei stiffer than the rest of the cell. This would be the apical surface where the cells are not adhered to anything, so they're bulging out a little bit. But here there would be adhesion to a basal lamina to the rest of the tissue, so it's flat and it's scalloped here at the cell ends. If you stretch tissue either this way or this way, you're going to naturally be stretching the inside, and you're going to get, it's not just in the same direction as the stretch that you've got stresses. There are localized stresses around the nucleus. If you compress the tissue, you're not just compressing in the direction of the compression tangentially, you are also getting some apicobasal stretch in the perpendicular direction, and you're also changing the shape of the free surface of the cells. You're bulging them out and pressing those cusps together. So maybe it has something to do with seating junctions. I don't know what.
So which material law do you use for this?
Linear elastic. Well, finite deformation. But it's simple.
But I mean, given the polymeric nature, I mean, I would
This is a
expect
start.
something like,
It's just a start.
okay.
It's just to be able to say something about what's going on and frame the questions that you'd be asking in the experimental system. This is not even the good stuff. Okay? So maybe there's just too much here. There's all these different directions, all these different locations. What else can we say? So there's fluid here. And how does the fluid interact here? And how do the dynamics come in? If you need to consider the dynamics, you got to get a little more sophisticated. And so we made a fluid structure interaction model where these elastic solids are interacting with the fluid. So we get to see the fluid structure interaction. And we pass a wave through the smooth muscle. And a movie is worth 1,000 words. And we actually have some estimates on the parameter values, but really it's just order of magnitude because it's such a widespread phenomenon. We're talking about a lot of different stages in development and a lot of different organisms. So really a range of stiffnesses and lumen fluid viscosities and so on would be worth considering. Now this movie is not, this is a jerky movie because Windows. So I'm going to, or because of Office. So I'm going to show you the movie externally. So it will not be so jerky. If the end is closed, the pressure is uniform everywhere. And it won't close completely because there's too much resistance. If the end is open, the fluid can drain, and you get the pressure build up only in front of the wave. And then a negative pressure as it's refilling. And I gotta stop this because that's later. You'll have to wait for that. It's not much later. So different things are going on if the trachea is open or closed. And we can use this model frame. Once we've developed the model, we can use it to measure the stresses on the tissues in different places, do a parametric analysis, which we did, and see how the stiffness and the fluid viscosity and so on affect everything. And this is just holding still in case you wanted to see the quantitative difference over time. Again, the red is a little bit dim for some reason.
How do you implement the contraction in
It's a body force that is meant to be circumferential, but because we're using an axisymmetric formulation, we actually model the circumferential stress as an inward normal stress, radially directed stress. It's a body load.
Is it the same like doing thermal elasticity?
Doing what?
Oh, if you lower the temperature, you have contraction. Would that be the same?
I don't know. This is not even the interesting stuff. Okay, so what's happening to the stretches in the cells? As in our simpler estimates, you get compression in one direction in the stalk and compression in another direction in the tip, and we can compare what's going on with the lumen open or closed, and we can also then ask what happens, does this depend on viscosity? And practically all of these cell stretches are not dependent on viscosity, but only on the ratio of essentially how much force to how stiff is the material. Some of the stretches, the ones with the tildes on them, are dependent on the viscosity of the fluid, because remember, when the lumen is open, it drains, and how fast it drains depends on the viscosity of the material. And almost all of these relationships are linear, but some of them marked with this are nonlinear. But that's just bookkeeping. We did a large parametric analysis. But there's one other thing to think about besides the stretches of the cells, there's flow in the lumen. Could the tissues be sensing that flow? Let's look at it. How much flow is there? Well, you can estimate how much flow there is. And think about it. If the wave is moving this way to a closed end, the fluid's moving this way. Right? I mean, you don't think of that, but the fluid is moving in the opposite direction from the wave. And how much? Well, it doesn't take much occlusion to move quite a lot of fluid in the opposite direction. And these are just estimates from conservation of volume and estimates from just simple pipe flow of what that pressure would be. And we're going to use that in a moment.
I mean, you probably solved this with zero flow boundary conditions.
Yeah.
Yeah, but this is exactly where the cells have to sense it.
Yes, well, in fact, that's what this slide is about. This is a plot of the midstream velocity and the shear at the surface nearby, and they pretty much exactly track each other. So if there is shear sensing, it would certainly be by, say, cilia on the surface. And that's a good surrogate measure for how fast the midstream flow is. So that could be that would be a reasonable mechanosensing location and mechanism. But think about it, there isn't actually any flow distal to there's not much flow distal to the smooth muscle. So you wouldn't expect to find shear sensing distal to the smooth muscle. And I would bet that you will not find primary cilia looking for shear distal to the smooth muscle, which is kind of cool. It's something that pops out at you once you start analyzing this system. Even though the models are very crude, you start to see things. One of the other things is we can just track where parcels of fluid are going, and I don't even want to take time for the movie. There is a movie of this, but if you notice, so the fluid is moving or is circulating inside the tube, and if the trachea is open, it goes way outside of the lung. There's stuff that exits and then comes back in. And that makes me think about mixing. So perhaps there's cell stretching that's stimulating the system developmentally. Perhaps there's flow sensing by primary cilia. But, you know, there's stuff in the lumen that perhaps is being transported and might be morphogenetically important, possibly. So, and a lot of this has to do with viscosity. So we did a little bit of rheometry on the mouse lung. Isn't that cool? I was so proud of him. This was at Farsh-Gilek's lab, which is about to move to the Midwest. We injected beads and tracked the random walks. And interestingly, it is Newtonian, and it is a little more viscous than water, but it is not nearly as viscous as mucus. Okay, so now we know something about embryonic lung fluid. It's not mucus, basically, but it is a little thicker than water. And we can model it as Newtonian, because very different things would be going on if it was more like mucus. But let's think about the transport. If there was no peristalsis, if you blocked it, it would take a long time to move chemicals from one end of the lung to another, particularly as it was getting bigger, if there was no peristalsis, or no movement of the fluid at all. I mean, later on there will be these fetal breathing movements, but it could take days to get proteins from one end of quite a small lung to the other. So what peristalsis is doing is it's adding advection to the diffusive process going on in the lung, and when you've got those processes going on together in a pipe, we call it Taylor dispersion, which dramatically enhances the mixing of the material of the whatever's in the lumen fluid. So it could perhaps be a useful mechanism just for mixing stuff up in the lumen. I don't know. There are a lot of things that it could be doing.
The importance of that, sorry to interrupt, depends on the Peclet number being significantly larger than 1, although your flows, I suppose, the Reynolds number is significantly less than 1.
It depends how much occlusion you've got. You can get so if you think about this, The closer you get to 100% occlusion, the faster the midstream flow is, because you're building up a pressure and forcing it through a small nozzle.
But I mean, in those embryonic lungs, is that enough to give you a Reynolds number greater than 1? Because the size is so small.
When you get towards 100% occlusion,
And the viscosity is quite a bit bigger than water.
it's only 10 times as big as water.
Yeah, exactly.
Yeah.
But this is true for laminar flows, right? Taylor dispersion.
This
Yeah.
formula is, I mean, Taylor dispersion
Right.
is concerned with turbulent flows as well. I mean,
So I thought this is for proper flows.
We are waiting for you to say that this is not the interesting part.
yes.
It isn't yet the interesting.
which is
That's on the
we
next
have a
slide.
Reynolds.
Yeah. So
But even so, it requires the Peclet. In other words, the diffusivity of whatever it is that might be being mixed has to be a lot smaller than the kinematic viscosity of the liquid for this to become important. And it probably is. It's not a critical question. I'm just asking what the Peclet number was. Work it out with our team.
Right. I'd rather do it later, after the talk, because I want to get to the interesting part. I realize this is interesting to you, but let's talk later. Anyway, airway peristalsis, you learn a lot just from throwing basic models at it, but it's still, there's too much going on. It's not a simple enough system to understand. I like those otic vesicles. They're cysts. They're geometrically simple, at least. But let's at least get something without a lot of dynamics, okay? If you stop up the airway, it matters mechanically, and it's a simpler system. If you take that mouse lung in vitro and just grow it normally with the trachea open, it branches nicely. If the trachea is blocked, in this case by cauterization, it doesn't shut down the growth. In fact, it enhances the growth. You get an increase in mitosis, and you get a dramatic increase in branching. Now, why would that be? So, the belief by the researchers who did this is that the increase in pressure stretches the tissues and causes more branching. To me, there's got to be a few steps in between the beginning of that sentence and the end of that sentence. Okay, so really what is going on? And to me, that doesn't really make sense. Okay, so this is something vaguely lung-shaped, right? It's hollow, and if you've got pressure, can I use your... Oh! I'm not buying that brand again.
Dangers of positive pressure inflation for the patient.
Yes, actually, in fact, occluding the airway was investigated as a method of reversing lung hypoplasia, but it was found to be injurious, so they aren't doing it anymore. If you think about it, you blow this thing up, it doesn't get more branched. It gets rounder. It gets rounder, right? It does not get more convoluted. It's mechanically it doesn't make sense. But I needed to find a clearer way to say that than a rubber. Actually, that's probably a fairly clear way, but we don't really know. The lung is not a simple latex shell. But so, what could be going on? It could be that there's stretch sensing that is leading to some increased morphogenesis somewhere. Is it even in the right place, for example, that we can throw a model at? Is it even in the right direction to be stimulating additional growth in some place? So we throw a model at it. Again, it is nice to simplify the geometry as much as you can and start with a simple tube. And in this case, we're just looking at a pressure vessel model. This is the work of my recent postdoc, Udo George, who was a student of Untitled and is now on the tenure track. And so if you think about it, occluding the trachea makes any secretions back up, and there's going to be a pressure in the lumen. And so we just track how much this tube is. It's a pressure vessel. This is a standard engineering task. And we can look at the stress at the different layers of the tissue, and not surprisingly, the highest stresses are on the inside surface and decreasing as you get towards the outside. And also not surprisingly, you have greater stresses in the stalk than in the tip area, because the circumferential stress is roughly twice that of the longitudinal stress. So if you think about it, the stimulus for stretch sensing would be in the stalk. The epithelium has its greatest stress in the stalk region, which is not where it branches. It branches on the tip. Okay, well, what if we have the real geometry? This is not the real geometry. This is just a straight tube. Is that true also in the more natural morphology? So we had to actually capture the natural morphology and take somebody else's stack of an embryonic mouse lung, segment it and mesh it to be able to do the same kind of analysis of where are the stresses if you block up the trachea. And this is a slice through the 3D. The simulations were done in 3D. And the colors are not right, but you can still see that the highest stresses are near the surface, or if you're thinking about the epithelium, the apical end of the epithelium, the apical surface of the epithelium, rather than the basal surface. It's really kind of hard in 3D to tell the details here, particularly with the slice. So another way to look at this is to just open it up and look at the stresses on the various surfaces. So this is on the mesothelial surface, and this is on the basal surface of the epithelium, and this is on the apical surface. And you can't really see the location of the peak stresses on each layer unless you rescale the stresses. If you think about it, different parts of a tissue would have different sensitivity to any kind of stimulus. You wouldn't necessarily expect that any kind of sensing on the basal surface would be the same, would be calibrated to the same level of stress as on the apical surface. But again, if you're expecting branching to occur at the tips, and it's sensitive to stresses, why is it that the peak stresses everywhere are away from the tips? Even on the basal surface, where you have some peak stresses on the tips, it's on the wrong part of the tips. So if the branch is going to happen here, the stress is in this direction. So with the glove, is this the broken one or the good one?
That's the broken.
Thank you. No, this is the broken one. This is flatter than this. And so when you inflate it, it's making it more like a sphere. It's round, it's bulging out the flatter parts, but the branching occurs on the shoulders, not the flatter parts. It's in the wrong spot. So stretching the cells doesn't make sense as a mechanism for the enhanced branching. But I had another idea, which I wouldn't have had if I hadn't been thinking about peristalsis for a couple of years before that. What about the transport of dye? Where's Kishore? Oh, there you go. Okay, of dye, of morphogens. We know a lot of the morphogens. So this is the interesting part. Okay. And this is going to run over because you had too many questions.
All right.
So this is just a 1D model. There are a lot of morphogens in the system, but I want to just keep it simple to start with. I want to start simple. That's Lubkin's motto, start simple. And just an imaginary morphogen representing a bunch of known morphogens. We know how they're interacting pretty much, but just if you have a morphogen produced at the pleural surface and it is transported to the epithelial surface, the basal surface, where it binds, I know there are morphogens that are produced in various places, but this is just kind of lumping them all together. If it is diffusing and advecting, think about it. So this tissue, there's fluid being secreted. We had the perfect preparatory talk. This fluid is being secreted into the lumen, and it has to come from somewhere. It's coming from outside, right? And it's passing through, it's passing through the tissue and binding. And depending on the Peclet number, you're going to get different profiles, either very flat or very linear. And as you block the trachea, you are slowing down the flow and getting a more linear profile. Okay. Okay. How does that tell us anything? Well, when you're slowing down the flow, you're getting a higher peak concentration in the mesenchyme, which you can get analytically, which is kind of nice. But what does that do in a real geometry? So we threw another simple axisymmetric model at it, where we didn't even consider the elasticity, but simply considered the epithelium and mesenchyme to be porous media through which the morphogen advects and diffuses, coupled to a Stokes fluid in the lumen, with normal flow, literally normal flow, being driven by some pressure gradient across this surface. And I don't know what's driving it. Can you tell me? Maybe not right now, but at some point. There's some epithelial secretion, there's some pressure gradient. I don't know what's causing it. But it's pulling the fluid with the morphogens in it. And when you occlude the trachea, you're not decreasing that pressure gradient so much as opposing it with the pressure here. So the flow stops. And again, we have some parameter estimates. We have a range that we're thinking about. What you see in 1D, you also see in the axisymmetric model, that when you block the trachea, you're reducing the Peclet number, and you get a higher concentration of your morphogen in the mesenchyme. So that kind of makes sense, and that's actually what they see in these systems for example, FGF10, which is, I believe, produced in the mesenchyme. You see more of it when you block the trachea. It also gets redistributed. They noted that you see more of it further from the tips, deep in the organ, further from the tips. They noticed that they see more of the FGF10. Okay, so that confirms what was seen, but it still doesn't explain why you're getting branching. When we throw the same model at the 3D geometry, and these are slices, you see basically the same thing. You get a higher concentration in the mesenchyme and more of it appearing away from the tips. But I still fail to see how this would stimulate branching at the tips. Because concentration in the mesenchyme is kind of the wrong quantity. You can image it, but I think you're not measuring the same thing. I think you're not measuring the right thing. And I think the right thing to measure is flux. If it's being produced at the pleura and all of it is being bound at the epithelium, then the flux is conserved. Everything that's being produced here is bound here. In 1D, that's not terribly interesting. In the axisymmetric case, the concentration is very different in these three cases. But again, the total flux is the same. Whatever is produced at this surface binds to here, and so the total flux is the same. But when you've got space in more than one dimension, you get a redistribution of the flux to different locations. When the trachea is blocked, which is the solid curve, you get a tighter distribution on this tubule. And then when you go to the real lung geometry, here's where it gets dramatic. This is the cool stuff. Okay, I promised you I'd get to the cool stuff. This is why I was hurrying you. This is the really cool part. So the streamlines are the same. That's not terribly important. What's different is the flux that arrives at the epithelium. The total flux is the same. If the trachea is open, it's in a few big regions. You'll get a few places of branching when this arrives and binds. As you close off the trachea and reduce the Peclet number, you've redistributed the flux to more locations. Now that makes sense as to how you could be increasing the branching rate. So just to review, what you see in 1D and axisymmetric, you also see in 3D that when the trachea is closed, you get these tighter locations of high flux, and it's in more places. And that makes more sense to me. And when I showed it to the guy who did those experiments, he was like, well, he liked it. That is literally what he said in his one-line email from his iPhone. That it absolutely made sense that you were spreading the morphogen to more places by blocking the trachea. And it wasn't mechanical. You don't need mechanics to do this. This is a transport problem, not a stretch problem. Oddly, and this is not a big thing to me, but it was super cool for him. We saw triple points, which would lead to trifurcations, which are kind of the stuff of legend, like fairies and ghosts and stuff. But he deliberately left trifurcations out of several publications, even though he sees them regularly, because there were reviewers who were saying there is no such thing as a trifurcation. But you can get it from the fluid flow. So to tie it all together with the static problem, yeah, there is mechanical stretch, but it's probably in the wrong place to be enhancing branching. Except actually, it could be in the pleura that there's some stimulation of release of FGF9, which then leads to enhanced release, enhanced promotion of FGF10, et cetera, et cetera. My point being, though, you can see concentrations, but you can't forget about flux, because that's the binding. And this weird thing happens when you change the Peckley number. It changes the distribution, and that is the cool part. So this is work with David Warburton of Children's Hospital Los Angeles, and Ed Jessidason, who's up in Liverpool now, is the peristalsis expert, and my terrific grad students, Oswaldo Lozoya and Kishore Bukka, not Mosley Ganti, and Uduak George. Thanks.
Thank you for this beautiful talk.
So if you were to include growth in this deformation,
Yeah.
do you think you will compensate for the flux with the deformation?
I don't know. That's too hard. I mean, this stuff was challenging enough. Growth, adding growth, you could. It would take some very sophisticated finite element methods that could handle things going on surfaces as well as in the bulk.
Sean?
You'll probably hate me for asking this, but I'm wondering about, there's a lot of assumptions in the model, so I'm wondering about effects like turbulence or cilia beating around and how that changes the results. And as an experimentalist, I would say, what happens like if you just inject dye into one location? Does it give you the flow patterns that are predicted by the model, or have you been able
Nobody's done
to
it.
convince somebody to do that experiment?
Deep fish don't have lungs, do they?
Unfortunate.
But they have swim bladders.
So one thing they're
They
bad
have
at,
swim bladders.
they're pretty simple though.
But well, yeah. So it's a different system. It's not looking at branching, but the tissue types are probably very similar because the lung evolved from the swim bladder.
Yeah, but no branching in this one way.
No, but you could maybe look at the pumping.
The last question?
Well, do you think there's turbulence? Or is it too small of a scale for turbulence? Or do you think cilia are changing movements of water? Or?
Not at this scale, I don't think. No. I doubt it.
Last question?
So can you also revert the effect of occlusion by injecting or removing FGF10? Because I mean,
Yes.
I would say
Yes. It's in that same paper, actually. The paper
Oh, right.
that.
so that's consistent with your model.
Yes, it requires FGF10. If you block FGF10
So
activity,
where
then
again is it produced? Is it produced all over the pleura, or
FGF9 is produced in the pleura, which is transported into the mesenchyme, which produces FGF10, which binds to the epithelium, which then releases sonic hedgehog. And there's a lot. It's a lot. I don't like networks. I don't like Sorry.
But I just missed where exactly in the model it is produced.
In the model, the morphogen is
Right.
produced at the pleural surface and binds at the epithelial surface.
Okay, all right, okay, yeah,
But representing a cascade of events.
right, of course, yeah.
Yeah.
So I think we should continue. So let's thank Sharon again.